Good day, everybody. So today is one of our important day. That is Professor Lalitha from India. Yeah. So you introduce yourself first.
Hello, good day to everyone. This is Dr Lalitha, Assistant Professor, Department of Botany and Soil Biology and PGPR Lab from India. And my specialization is Agriculture Microbiology. Thank you.
So Dr. Lalitha is visiting professor and also she is doing research with us. So here we are at the Research Institute of Modern Organic Agriculture, or LIMO. Belong to King Mongkut Institute of Technology, Lattabang, Thailand. The topic that Dr. Lalitha will give presentation today to all participants is about microorganisms that can degrade heavy metal, because this is one of requirement to do organic agriculture. So right now, today, not only Thailand, I think every country all over the world, we concerned about the organic product, organic agriculture. That means we have to stop to apply all agrochemical. So after we stop to apply on agrochemical, some country or someone, they try to do it, but they not really success because of they do something like traditional organic agriculture. So the traditional organic agriculture sometimes they cannot get the yield, both of quality and quantity. Maybe cannot enough for yield or for survival. But what we do is the modern organic agriculture. That means we will introduce, we will recommend all the farmer they are do organic by scientific research, investigation to do. So that means this is we call modern organic agriculture. So that means we are a group of the scientists working in this area, not only from LIMO, KPTL, but also we have consortium or collaboration with many countries, like India, Egypt, Philippines, Laos, Cambodia, Myanmar, Turkey, China, Iran, and USA, also USA, Germany, yeah, so many we are working together. So today is a very good chance for all participants to register for the online international training for organic fruit production in tropical climate, and also integrated organic farm for organic fruit production. This is an important point that I like all participants to learn, to know about this. So before we are going to let Dr. Lalitha give presentation, I would like to invite our Vice President of the King Mongkut Institute of Technology to give a talk and also opening our meeting. Yeah, please.
Thank you, Dr. Kasem, for your introduction. Yes, and good day, everyone, and welcome to the international training on organic food production. So, this session topic is related to the reduction of the heavy metal, right?
Yes.
Which is one of the key parts.
Yeah.
I and Dr. Kasem actually share the same goal. We would like to reduce the cancer in the world.
Yes.
And heavy metal is one of the main factors. Yes. So it would be a lot of impact. If this kind of knowledge can be spread around and be utilized in every part of the world. Yes. And today he introduced me, Dr. Lalitha.
Yes.
He said, "Oh, this is very interesting."
Yeah.
And when he said interesting, I believe in my heart that it should be very interesting. So I would like to welcome Dr. Lalitha and thank you for giving us the special lecture today. And then for everyone watching this, I would like I think you will have a good time and good content and good practice from this lecture. So I would like to open the special lecture now.
Okay.
Thank you everyone.
Thank you.
Sawasdee
Thank you very much.
Thank you.
Please stand by here. So the Dr. Lalitha is do collaboration with us over 10 years, almost, and he did a very excellent. Now today, Association of Electronic Technology in South Asia or ASE win award to him. Dr. Song, you can give award to him. Let the Vice President give award. Research excellent award to Dr. Lalitha. Come here, we give you Vice President. Okay.
So
Yeah. Okay.
Thank you.
Yeah.
Thank you.
Big hand. Congratulations. Congratulations.
Thank you. Thank you. Thank you.
Tại thăm nhân nha. Important.
Yeah.
You be inside.
Yeah. Because the,
okay.
yeah.
Okay. Okay. Congratulations. Congratulations.
Thank you.
Okay.
Thank
Thank
you.
you. Thank you, my president. Thank you. Okay. You can start.
Yeah. Good day to everyone once again. And today I'm going to discuss about the innovation on soil remediation and reduction of heavy metals by potent microorganisms. And myself, Dr. Lalitha from Department of Botany, Soil Biology and PGPR Lab, Periyar University, Tamil Nadu, India. Hope another 1.5 hours you may be explore on heavy metal toxicity, how it is going to impact our plants, animals as well as in the human, what is going to give the remedy. And first, we have to think about what are the main sources of heavy metals in agricultural soils. So if you think where the heavy metals are coming from, so you have to think about how it will deposit on the environment. So if you take the environment either in the soil or in the air or in the water. So where is the heavy metal may come from? So before that, what are the heavy metals? How we have to say what are the heavy metals? The heavy metals like cadmium, lead, arsenic, chromium, and mercury, copper, zinc, and nickel. So these are all the heavy metals. I think you may be aware of these heavy metals. You can see every day using the automobiles or anything in the transportation. You may excellently you may explore the lead and cadmium, which is emitted by the automobiles. Okay, and apart from that, vehicles, you may get this heavy metals from mineral fertilizer, which where you have to apply on the surface of the field. Any field, if you take this mineral fertilizers, if you are applying, definitely you will get a chance of these heavy metals and it will deposit on the atmospheric deposition. So if you have the chance of atmospheric deposition, already the molecular nitrogen is also there, and along with this mineral fertilization, explore on that field, it will deposit on the atmospheric deposition. And then another one, it is a very deadly dangerous sewage sludge. This is the main reservoir of the heavy metals in the pollution. One of the major reservoirs is sewage sludge. You will get all kinds of that is microbes and heavy metals and whatnot. Because this sewage sludge not only from the domestic as well as also the industry. You can have it either the paper industry, leather industry, or in the capsule form, in any way, even dyeing industry. Dyeing industry is the worst one, where you could see the all kind of heavy metals. And another one is the pesticide spray. So if you have the pesticide spraying in the field, either in nowadays we are talking about more about the drone or in the spraying in the foliar application. Every day we are improving in the that is the sophistication of life, but we forget what is the impact on the sophistication life. So drone spraying or any method of spraying in the chemical way of pesticide, which will give the highly impact on the, that is the high impact on the heavy metals which is deposit on environment. And then sewage irrigation. We are talking about the sewage irrigation by means of, you can that is purified, and you have to clean up the sewage and give it to the field. It's no problem. It's appreciable one. But what about the heavy metal degradation on the after replantation, after treating the sewage water? Have we checked out? Not yet. So that after cleanup, after treatment of the sewage water irrigation to the field, it will pollute the soil as well as in the aquatic environment. And then the major thing, if we start to organic agriculture, first in our mind, are we in the rural background, first in our mind, what do you think? We can apply the manure. Oh, manure is good for organic agriculture, yes, of course. But the source of manure, the plants may eat the fodder or any other greens which may contain the heavy metals, cannot degrade by the cattle. So that deposition, where it goes into the manure only. So in that manure, have we ever checked the manure may contain the heavy metals? No, not yet. But we are recommending the manure, it's a good for the organic way of agriculture. Yes, absolutely true. When it is the organic cultivation field, that fodder given to the cattle, it is a good for health as well as in the healthy manure free of heavy metals. So nowadays, even the scientific growth, it is going up. We have to think about these things, how the heavy metal is polluted. Now in my session, I have segregated in 3 parts. One is what are the sources of these heavy metals are from, and then how the plants may affect these heavy metals, and how to give the remedy through the microbes, and what are the impact on the human, and finally what is the solution we are going to give, and how to rid it off the heavy metal from the environment. So we can go by the segregation. And then next we can see here, first topic, that heavy metal stress response to the plants. It is the most important. We never think about the plant and its growth. We have to concentrate our own growth, because we know if we don't like the food, we can refuse. We can say it by the language. So in plants, what it do? Only thing is it won't grow. If there is a not in favorable environment, the plant won't grow. So that is the response by the plants. So we should understand that the plants may not have the good environment which cannot be grow in the particular soil or in the water. So for that, what are the indicators how the plant responds to the heavy metals or undesirable environment? environment. First of all, if you take the two parts, growth and correlated with the physiology. So if you think about the growth, what is the first sign? Germination inhibition. It won't grow. The plant won't grow if you have the plant soil with the heavy metals, right? And second one is the growth reduction. It will germinate, it will grow up to 5 to 10 centimeter. After that it won't grow. And then second is premature senescence. So what is premature senescence? The same thing. It will grow and die. It will grow and it will become a senescence. Green, yellow leaf, and it will fall down. And then reduction in productivity. So even though after this tree it overcome to grow, it won't give the production or it won't give the yield. So how this physiology, superficial growth will correlate to the physiology. We can see to do that. Reduction in water uptake. So automatically either all the living organisms, those who cannot uptake of water will become shrink. The plants also cannot afford the uptake of water, become shrink, become old. That is we call as the senescence, right? And then alter the transpiration and the respiration rate. So the plants, the vital part is the transpiration and the respiration. If it is transpire and respire, the plants can survive. So for the plant's survivability, the both are very important, but it will stuck by the heavy metal induction, and then it will affect the metabolic toxicity. So what is metabolic toxicity? Metabolism meant by both the catabolism and anabolism. That is the breaking down the molecules and you have to combine the molecules. So you have to generate the food and transport the food into the root to top leaf. If it fails to do that, the plants cannot grow. And the accumulation of growth inhibitors. This is the pivotal important important factors. If the growth inhibitors stuck, the plant will grow. So what are the growth inhibitors? I will detail in the later slides, because if you want to grow the plants, it grow upward and downward, both the aerial part and the below ground part. In that, what will happen? It stuck the auxin production. So it won't produce the auxin. Auxin invariably you can talk about the IAA, indole acetic acid. So if cannot produce the indole acetic acid or by means of the auxin production stop, the plant cannot grow. So that is where now you can correlate that physiology and the growth. So the physiology, reduction in water uptake is directly related to the germination of inhibition. Growth, germination, inhibition. So that it won't be grow. The seed, if you sow the seed, it won't be germinate. We have to wait a month together. It won't be germinate because of the heavy metal deposition on the soil. Okay. And second, alter the transpiration rate and the respiration. So growth reduction. So you cannot get the right growth at the right time, because you may not have the transpiration and the respiration rate. Again, it's stuck by the heavy metal. And then metabolic toxicity. So if you cannot have the metabolism occur, so what will happen? Automatically it will affect the premature senescence. So premature senescence will happen only of the metabolic toxicity. And then finally accumulation of the growth inhibitors, it reduction in productivity. So you can correlate with the growth and physiology is both is directly connected, and you can cross the plant what is going to inside the plant, this physiology may occur due to the over accumulation of the heavy metals. That's what that heavy metal stress responds to the plants. You cannot get the proper germination, growth, and it goes to the premature senescence. Okay, and then next coming to the previous slide we discussed about that is the physiology and growth. So what will happen to the inner part, that is the internal? What will happen to the proteins and gene, that is the omics? We are dealing about the omics, proteomics and genomics. What will happen to the plant? It affects the proteins associated with the cell wall growth. So if without the protein is the building block of the entire health system. So if you don't have the proper protein development, that cell wall growth won't be happen. Okay, and then next, decreased expression of cyclin dependent kinase. What it is mean? Kinase, if you have that kinase, you can directly correlate it with the ATP synthase. So ATP synthesis won't be happen. You cannot get any energy. So the plant will die automatically. So the decreased expression of the cyclin dependent kinase will affect that is due to the heavy metal impact. And next one is the affects both elongation of meristem zones by altering the auxin distribution through the P1N1 protein. So already I told you about the previous slide. So growth inhibitors. So what are the growth inhibitors? In meristem you have the auxin. In root the meristem you have the gibberellin. So if these two may stop, the plant won't grow. So directly it is related to the auxin production. So in all meristematic cells, either in the nodal part or in the upper aerial part, wherever the tip of the plant system which is responsible for the growth by the sideway or the upright, you can have the auxin. So this auxin production abruptly stopped by the heavy metal. So what is the P1N1 protein? I will give you later. And then targets of transcription factors and sulfate transporters. So here transcription factors, if you fail to do the transcription, you cannot get the gene transfer, that is translation won't be occurred. There won't be transfer the genetic material from one to another, F1 generation to F2 generation. You cannot transport the genes, that is the genomics. It heavy metal drastically affect in human as well as the plants, drastically affected the genomic level. That is primary, it will target the transcription factors. We knew about what is transcription and translation. It won't be transcript, you cannot get the genetic material transfer from F1 to F2 and then sulfate. It is another genomic important factor chemical and which may not be transport. It will be stuck by the heavy metal, then altering expression of microRNAs. You know mRNA. mRNA is the major RNA which will transport again genetic material. You want to transport the genetic material, you need these things. So you need to protein to transcribe and the gene material has to transport. Then only you will get the second generation. Otherwise you cannot get it. So these major impact will happen by the heavy metal. That is the major impact will happen to the heavy metal deposition in the soil, air or in the aquatic system. So we have to keep it in your we just ignored the heavy metal impact on the environment. So nowadays, every day we are growing. In scientific way we are growing. Advanced technique in modernization due to the hidden prices of the heavy metal increasing every day. So we are ignoring. We are talking about the pesticide, herbicide, everything in the broad spectrum. But what about the heavy metals? These heavy metals, why it is very dangerous? In the low amount, in microgram amount per liter, it will cause the deadly dangerous effects. So there is no remedy at all. That's what we have to be awakened. So what is that? Another, are we going to give this one for the chemical source? No, because already using up of more and more chemical only, we are in the position to increasing the heavy metal in the environment. So in that continuation of the previous slide, you have to that response of mechanism rhizobacteria in the heavy metal impact. What we are going to do? So what is the impact? It increases the action of stress related proteins and signaling molecules like calcium ions and ROS in various hormones and MAPK, that is nucleogen elevated protein kinase signaling cascade, transcription factors, that is basic leucine, zipper, WRKY and ethylene responsive protein, heat stop proteins. So what are they? So in previous slide we saw the impact of the heavy metals on the plant. So if you are giving some rhizobacteria on working on the heavy metals, what will give the impact? For example, Bacillus subtilis, Pseudomonas, Citrobacter, Bacillus and Pseudomonas acarifa and Pseudomonas aeruginosa. These are some of the microbes, bacteria which we worked on the lab and tested, and these are some of the bacteria which act upon the heavy metal and give the stress related protein, that is induced to the plants, act against the heavy metal impact, and it gives the signaling molecules like calcium ion. It stimulates, it induces, that is the calcium ion, you know very well, in human system as well as the animal system or in the plant, calcium and sodium and potassium. These are all the stimulators. You can ionize, transport these three ions only. So you have to transport. You need these ions. So gives the signal to the calcium ions. Yeah, you have to awake, the heavy metal is coming, and the molecules, microbes will awaken, give response immediate to the plants. And then mitigation, aviated protein kinase signaling. These are all some of the secondary alerting green, red signal to the give to the plants. Then the plants is alerted and it escape the heavy metal impacts. And what will we do? What is the action? These microbes onto the plant. It gives the low molecular weight organic acids. Example, glucamic, oxalic acid, citric acids, which play positive roles in the mobilization and solubility of heavy metals. Okay, and then biosurfactants form complex with heavy metals in the soil surface, dissolving metals from soil matrix, and siderophore are the siderophores which are able to bind metals and thus enhance their bioavailability in the rhizosphere through a complex reaction. So these 3 points we go one by one. How the low molecular weight of organic acids will produce. These microbes will produce the glucanic acids or citric acids, which will break down into the complex to the simple ionic form, which may easily dissolve in the air or in the water. So if it is the complex one, it will be harmful to the health. So you have to slowly reduce their action or reduce their negativity. And biosurfactants. What is the biosurfactants? This biosurfactants is nothing but it form a balloon-like structure. So it gives the elasticity, it gives the balloon-like structure, and it acts upon the ions, and it bind with the microorganisms, produce the biosurfactants on the surface of the heavy metals, and it protect the plants. The heavy metal won't bind or won't go into the inside the plant through in the rhizosphere. Okay, I will give you the detail later. And the siderophore. This is the very good siderophore is the very good product which will produce by the microorganism, especially plant growth promoting rhizobacteria. All these things may produce siderophore. So this siderophore is nothing but iron chelating compound. So this is the iron chelators. So these iron chelating compound, it's a protein. Okay. So these iron chelators not only chelating means breakdown. So not only break down the iron which is non-available form in the surface of the soil, also it will chelate the other heavy metals which may not degrade by the plants as well as in the soil. These microorganisms produce the siderophore which may break down the heavy metals. So that's what we have to encourage in the plant growth rhizobacteria. Here you have to notice biofertilizer versus the PGPR. Here the biofertilizer which will give the that is the growth of the plant, whereas the PGPR not only gives the growth of the plant, also protects us. It is the savior of plants from the heavy metal as well as the pathogens. That is the difference of the biofertilizer and PGPR. So in previous slide we saw the P1N1 protein, right? Yeah, here we can, it both affects both elongation and meristem zones by altering the oxygen distribution through the P1N1 protein. So now we come to that P1N1 protein where it is located. So this P1N1 protein is located basal end of the vascular cells, which is responsible directly acropetal oxygen flow in the root stem, or oxygen transport has been proposed by to play a role in stress induced changes. For example, A1 inhibits root length by affecting axin redistribution via modulation of OX and P and N2. So you have to think about the growth where it is the meristematic growth. How this P1N1 is localized. You can see in the B picture there is the nodal growth as well as in the aerial part. So for that portion, this P1N1 is present, which will induce the growth of the plant. And then now we are coming to the heavy metal deposition in the plants. Where it is deposited, how it is deposited, and what we are going to do according to the deposition. So if you are taking the hyper and non-hyper accumulator, you can divide the heavy metal deposition in plants in two types. One is the hyper tolerant non-hyper accumulator, and the second one is the hyper tolerant hyper accumulator. So this is the general. The heavy metals present in the soil in two types. That is the below ground part we have to talk about. One is the hyper hyper-tolerant non-hyper-accumulator. Another one is the hyper-tolerant hyper-accumulator. So it will, these metals taken up by the plants in the rhizosphere. We know about rhizosphere. Rhizo is the root, sphere is the surrounding. So the surrounding soil in the root system, we call it as the rhizosphere soil. So in that rhizosphere soil, there are 5 stages accumulate the heavy metals on the root system. So the root taken up this heavy metal by the 2-way and it goes up to the aerial part. So in ground part over, now going to the aerial part. That metals movement from soil to above ground tissue. Where it is stored, either maybe the stem or maybe the leaf. Okay, so if leaf, less metals uptake by above ground tissue. Less metals. The leaf may not accept the heavy metal deposition in the leaf. So that is we called the non-hyper accumulator. Okay. In other hand, metals, more metals uptake by above ground tissue. Okay. So after the harvesting, you may have the chance of deposition of the heavy metals on the plant. So if you are consuming leaf, you may have the chance of the heavy metals on the leaf, leafy vegetables. So think about the heavy metals. So that is more metals uptake by the above ground tissue. We can call it as the hyper accumulator. So it may be remain in the leaf system. So for that, what we are going to do, this kind of system, that is the accumulation in the foliar region, again it will impact the plants as well as those who are consuming. That will have the impact. That is the heavy metal deposition on the foliar. So in that case, what we do? Translocation of heavy metals. First we have to saw the slide. You may have the chance of on leaf deposition. No. So what will we do? What is going to do inside? So the root system may uptake metals, heavy metals. Either you can take it as the nickel or copper, that is the cadmium, or that is the lead, or chromium, or zinc, arsenic, everything. If it leads to the above level, we call it as the heavy metal. So it is uptake by the plants. What it goes to the stem. So you know very well about the anatomy of the plants. In anatomy, you can go for the stem. You will have the xylem transport. So in xylem transport, the heavy metal through the vessels and tracheids, this heavy metal will pass on to the each cell of the stem in the plant. And what will we do? In that, in your left hand side, you get detoxification. How these detoxification may occur? So transporters will taken up the heavy metals and go into the cells. In that, inside the cells, you know very well, if the HM means heavy metals. This heavy metals goes inside the cell. Each plant cell, you may have the different parts, cell organelles, right? You may have the endoplasmic reticulum, vacuoles, and mitochondria, so on. Okay? And these organic acids, inside the organic acids, which will produced by the endophytes, which you can see. Yeah, thank you. You can see these vacuoles, this iron filaments, here HM goes inside through the transporters. You may have the cell organelles here. So if you have the endophyte, good bacteria in your cells, that is in the soil, it protect, it chelates. I told you, no, siderophores are also called the chelators. Also it chelates the heavy metals. So chelation will take place here by producing the organic acids. In the third slide, I told you about it producing the organic acids, siderophores. So it produces the organic acid, amino acids, and it gives the chelating HM complex. So what it means? Heavy metal may be chelated by your microorganisms, positive microorganisms. So you will get the chelated, which will deposit on the leaf. Now your leaf is safe. Whatever you have to consume the leaf, it may be having the deposition of heavy metals. It won't be affected. In previous slide, I told you the red alert. Now you can freely, happily enjoy the leafy vegetables because of our heavy metal microorganisms. So the impact of the heavy metal microorganisms. What I told you in the previous slide, bacillus and pseudomonas, which will help us to chelate the HM, even though it deposited on the leaf, stem, root. Because we know many of the rooty vegetables, that is rooty vegetables like beetroot, radish, carrot, everything. That is also deposition of heavy metals, right? Here the major source, then it transport less. So the below ground part, even red potato, yeah. So we can have everything, sweet potato, everything, what I consumed yesterday. So we have to afraid to consume. So here we have to think wisely. What it happen? So we are the safer zone because of our microorganisms. Heavy metal microbes will save us. Eco-friendly manner. Only in this world, microbes only save us. Either in the human gut, it starts from the mouth saliva to the end of. Okay, so the microbes play a vital role rather than anything else. That's what we have to treat the soil through microbes, not by the chemicals. That is the important, that is the main agenda by our ATSI. That is the main aim. 20 years ago, Professor Cassim Saita from King Mount University. I am not anything extra exaggeration. This is his own research because we are associating since 2007 onwards. That time I was a student like song. I just interacted with Professor Cassim. So that time he taught me how to isolate the fungus, how to isolate the bacteria, and what are the positive and the negative, which is beneficial, which is harmful. He taught me. Whenever I come and associate, he taught only about the organic way of agriculture. He 100% hate against the chemicals. Don't talk chemicals. He always insisting on. I don't bother about the huge production through the chemical. I want the human live long, live smiley way. So you should go with the organic way. That is what his motto. So that he started the association, ATSI association. And then it is joined by the KMITL. So that we are so happy that young generation, now he brought many young generation across the globe. Across the globe. Chinese, we have Song is there, Philippines is there, Japan is there, and Cambodia, Vietnam, all Asian people. He sowed the good seeds of young scientists, budding young scientists. In our India, we were the President of India, Dr. Kalam. I think he knows everyone. He usually give talk to the school children because the young mind will stimulate in a positive way. The rest of the people, that generation will grow in the positive attitude. We need not to teach about the aged people. We can talk, no issue. But the young people, chemical, you should follow the organic way. That's what I inspired by Hassan talk as well as his practical way. Yesterday he visited many organic farms. I personally, and now I want to explore myself. I thought, is he doing any chemical, small amount of chemical mixing and that? He doesn't do it. He thoroughly mixing. I started from his lab to land. I visited. He preparing the organic way of microorganisms and mixing the organic product and bulk mixing in the blue color big tank. And one of the students, scholar, here they call it as the researcher from Cambodia and another from Philippines. He is spraying that organic product to the entire field. That you feel the air, you feel the smell in that ground. I won't get any bad smell. It's that grass smell only I felt. That's what I realized. Yeah, this is the purely organic way. And soon after spraying, usually whatever we spray, even we are using the antiseptic now. In that after COVID, we are using the hand sanitizer. After that, you won't get any that is mites or housefly. But soon after he sprayed the entire field in his organic product, the birds are happily sitting on the ground. We saw nakedly and it enjoying that moisture and environment. That's what I felt Professor Kasim is doing, not only the human, also in the other living organisms, living things like birds, animals, cattle, because the cattle may eat the grass. Wow, how? It won't say, I am eating chemical. Okay, so it will pity, it may cry inside the intestine of the cow or whatever you take. So after using the organic way, it happily giving the milk, organic milk, it happily given the egg, organic egg. His intestine will bless us. Really, these are all I want to tell. This is not at all a promoting. This is since I have associated with 2007 onwards. So I am seeing every stage. We have traveled more than 13 countries. Wherever go, he used to say, see, this is organic way, this is chemical. Which part you like? Chemical or organic? Oh yeah, that's what. He stick on that. Nowadays, everyone is growing by inclusion of the chemicals. They become a millionaire, but he doesn't want. He used to say non-profit organization. I don't want the money. How you people are saying like that? I amazed it because I need money. He said, no, don't go behind the money. You serve to the human. You serve to the living organism. Oh, that's what I realized. That's what he has a student of in many countries. I think in this forum, in this audience, many of them admire Dr. Kessin. Then only you have to register. More than 60 countries people, now you are watching, and you people also agree with my statement. So now come to the point. So here, this heavy metals may translocate and it will detoxify on the leaf. So you can have the leafy vegetables with the peaceful manner. And next, heavy metal remediation. This heavy metal remediation, if you go to the heavy metal remediation, what are the ways you have to remedy? One is the phytovolatilization. You know volatile. Volatile means evaporation, right? And then phyto-evaporation. Then rhizome degradation. Then phyto-desalinization. Then phyto-extraction. And phyto-stabilization. Rhizome filtration. These phytoremediation. These things, first in that phytoremediation, first you have to see the phytovolatilization. What it is? It is uptake followed by volatilization evaporation of contaminants in less toxic form as a result of metabolic modification through plant leaves. So through plant leaves, the phytovolatilization will occur. In previous slide, I show you it will be deposited on the leaf. So it will be volatile and it goes off. I will tell you one by one. And then phytoevaporation, uptake of contaminants from soil by the plant roots converted into volatile form. It will evaporate and the subsequent release into the atmosphere. And then degradation. This degradation again and purification through plant root itself. In plant root itself, the degradation will occur. And then phytodesalinization. So this is the removal of excess saline soil by the plants. So salinity is another stress to the plants. Either may be any means. That is a different big ocean of study. So next is the phyto-extraction. So uptake of contaminants from soil through the roots, translocation and accumulation above ground biomass. Above the ground. Previous slide you can see here it will uptake, it goes in the above ground level. So above ground level it will deposit. And phytostabilization is the above ground level part deposited on the heavy metals. And rhizofiltration, it filter itself in the root system itself. There will be uptake by the roots. Okay, and then first phytovolatilization. So in phytovolatilization, below the ground part, best example is the phytovolatilization arsenic and selenium. This is I am giving the two examples, not exactly all the heavy metals or any other heavy metal. Maybe there you may search and you can do it, right? I am just giving the example. In below the ground soil part, the gas exchange will be taking place and uptake of pollutants by the roots. Okay, by the roots, and indirectly phytovolatilization will be occurred, and in the above part the air and phytoevaporation will be taken place, and this heavy metals may to mixed with the air. How we to do it? By the sun. So when heated, the foliar region, the direct phytovolatilization will be taken place. The heavy metals may be mixed in the air. That is also pollutants. We cannot get rid of the pollutants. We keep it in our mind. The arsenic and selenium in the soil, it goes up and deposits on the foliar region through the sun, heat up, then phytoevaporation will be taken place and it mixed in the soil. Next, phytoextraction. So these are all the remediation. And again, this soil being remediated plant uptake by the contaminant. What are the contaminants here? That phytoextraction, the heavy metals like cadmium, arsenic, chromium, copper, lead and nickel. So these are all the chunks of heavy metals through the phytoextraction. You can remove heavy metal through the phytoextraction of these heavy metals. How? This contaminated in the soil, plant may uptake and the soil being remediated. How? That harvest in short contaminated taken up by the plant tissues. These plant tissues once deposited in the soil, that translocation in shoots, you have the biomass can be reduced by which? By means of compaction, composting, thermal transfers. For deposit, use the metals recovery. It is a phytomining. This is also doing. This kind of research is now booming in that scientific forum. So biomining, you have to harvest the plant which may have the heavy metal. You do the biomining. Here you can have the gold, mercury, expensive metals. You can have it through the phytomining. But it is a tedious process because microgram very less amount. But you have to do with the biomining. This is also one of the process. And then next one is the phytofiltration. How it goes. I think one of the research scholars is doing here, that is the hydroponic work. So it can be developed, hydrophonically developed the filtration, and the phytofiltration contaminants from the water in aquatic environment, that iron, nickel, zinc, copper, lead. These are all some of the heavy metals which may be in the aquatic, and the contaminants are absorbed by the roots. Then you have to remove the roots. This is one kind of phytofiltration. The plants may be filtered, the heavy metals in the aquatic system. You keep it in your mind. So the plant absorbs the heavy metals in that aquatic environment, they free from the heavy metals. So in the aquatic environment, the animals know snails or fish, it may free. So this kind of work is encouraging now. Those who are doing the hydrophonical, you may think about this work. You can incorporate it in your table, one of the research work through this. And next one is the rhizodegradation. So rhizodegradation, you can see the financial herbicides, plant secondary metabolites and metabolites, bulk soil bacteria, rhizobacteria, endobacteria. Here what will happen? Here you can have the heavy metal like lead, chromium. So lead, chromium may be deposited in the soil, in the rhizosphere, and taken up the plants, translocated in the aerial part. How it translocated? In the cells. So here our hero, who is the hero? Endophytic bacteria. What endophytic? Not only a bacteria, endophytic microbes, because arbuscular mycorrhizal fungi. I think many of them may knew this. This is again one of the plant growth promoting fungi, which is sporulated. So it having the spores only. This AM mycorrhiza, arbuscular mycorrhizal fungi, shortly we call it as the AM fungi. This AM fungi is an endophytic, and another our endophytic bacteria hero is the rhizobium. So rhizobium, which is endophytes and form one house or dwelling place, we call it as the root nodule. So in that root nodule, inside the plant cell, may enhance or may do the phytoremedial process, where in root itself. So in root itself, it rid of all the heavy metals by the endophytic bacteria and fungi. It rid of all the endophytes. So it cleans them and it goes to the in aerial part. It may be nullified 90%. Okay, so that is called the rhizodegradation. In rhizo itself, roots itself, the degradation will be takes place by the endophytes. It may be the fungi or may be the bacteria. Then representation of salinization mechanism in plants. So what are the salinization? Salinization you know very well, sodium and potassium. So in salt exclusion here and the sodium accumulation, you know the plant structure inside. You have the vacuole, cytoplasm, extraction, that is everything inside. Though the ions may be salt excretion, excess of salt may be deposition on the foliar, on the upside of the leaf itself. Sometimes you may observe there will be a chance of salt deposition on the leaf surface. So that kind of excretion may happen the over limit of the salt on the ground part itself. Because in ground part the root cannot do, with the help of the microorganism maybe the filtration or anything will happen. if it may be absorbed and it directly goes to the aerial part. So the aerial plant may be the salt excretion on the deposition of the leafy area. And then next, evaporation. So phytoevaporation. Here again you can see the nickel, lead, copper, cadmium, zinc. Here again the root system absorb all the heavy metals. There is no point that plant doesn't know this is a heavy metal, this is the thing. It recognizes when, if it is the organic soil, it recognizes, or else it is taken up everything. So what we do here, these heavy metals may be chelated, increasing metal solubility microorganism present in the ground level soil, uptake of metal chelatic complex by the roots, EDTA, and the translocation of complex in the shoots and converting into volatiles, releasing into the atmosphere. So now we may have the question, if it is releasing in the atmosphere may be polluted. No. How? Already we have the chelating microorganism in the ground level itself. It chelated. What is chelated? Breakdown. So complex heavy metal breakdown through the EDTA production, and these nickel, lead, copper, cadmium, zinc all will be broken down. Okay. So after broken down, it broke and it will take up by the plants. So after it may converted into volatile simplest form, it may be released to the environment. The environment won't be harmful by the heavy metals, because already here itself nullified. So that is called the phytoevaporation. Next, so far we have seen the heavy metal, how it will be degraded. Now what will be doing the plant growth promoting rhizobacteria or rhizofungi? So it will give the sustainable agriculture. How it gives the sustainable agriculture? What is sustainable agriculture? Long lasting agriculture. I saw while coming down to the airport to the city, I saw the quote, "Live long king." So the same proverb I used to hear, that is the long lasting agriculture. You go for the modulating phytohormones level and facilitating resource acquisition and crop production improvement, biocontrol of phytopathogens, and triggering induced systematic resistance. ISR. Professor Kasim often discussed with me about the ISR. You have to concentrate on the ISR, because this ISR have the triplet compound, triplet way to protect the plant, okay, and the healthy soil. So if you are doing all those things, you will have the healthy soil and you can achieve the sustainable agriculture. And then micro heavy metal interaction. So we saw the heavy metal and the types and how to promote the sustainable agriculture. Now what will be doing this microbe and the heavy metal? We are often talking microbes, you have to enumerate the microbes. Then what it will be do? What is the function? What is the action? Here you can see for the entire about that, I have given the detail about the phytovolatilization, rhizodegradation, rhizoevaporation. Everything here you can see the entire thing as a nutshell. So what it will do? The microbes, heavy metal interactions. First, what will be here you can see biopsin. Biopsin, what it means biopsin? Chelation, that is the breakdown. Coordination, complexation. Complexation means what it is? Electrostatic covalent with the EPS, exopolysaccharide. I will give you detail about the exopolysaccharide later. Then precipitation, reduction, cation, anion exchange. So these are all the points do by the microbes before the biopsy. Biopsy means bioabsorption, which simply called as the biopsy. Biopsy means what? Here, this is the heavy metal. So this heavy metal, before taking up this is the plant cell. You imagine this is the plant cell. So here, the heavy metal is there. So this heavy metal, before taken up by the plant or enter into the plant, this microbes, our heavy metal microbes will do action the chelation work, complexation work. Complexation work means electrostatic. That is the binding of positive and negative anions interactions. Okay, it catch hold are biosurfactants, and you may have the precipitation. It will deposit. It won't go into the plant. And the reduction and cation anion exchange. So after these process and enter into the heavy metal into the simplest form into the plant cell. Another what it will do? Bioassimilation. Bioassimilation means what? Bioassimilating. It itself degrade the heavy metal. So what it will do? It produce the siderophore or siderophore. So it produces the siderophore and it goes into the intracellular. Intracellular means within the cells. So you may have the complex heavy metals here, and it is bioassimilated by the microbes. It enters into the intracellular. Yeah, but it is degraded form. And another one is the bioaccumulation. You may have the active and passive transport, surface absorption. It stick on to the surface. So it won't be go enter the heavy metals. It will be degraded here itself and then enter into the intracell. After enter into the intracell, these heavy metals converted into the non-toxic metals. That is be achieved through the microbe. So this is called heavy metal degradation. So degradation means it won't be fully washed out. It is there, but how it is there? In the non-toxic metals, converted into the non-toxic metals, and then these things transformation. How? Through the biotransformation. How? Phytochelation, metal ions, and polyphosphates. I told you about these three may combined here, converted into metal ions. Oxidation reduction process will go off, and it will use the biomineralization. Biomineralization, if it is into the environment, it is a simplified form. Both oxidized soluble metals, reduced soluble metals, biological oxidation reduction takes place. It will converted into the simplest form. And here, bioprecipitation. I told you here now, bioprecipitation. So this bioprecipitation may converted into the organic acid, insoluble metals into the metal chelators, phosphate, metal carbonate, hydroxide, sulfate, which is available form to the plants. It's a good thing now. And here bio-leaching. So you have to leach out the complex form SO4 into simplest sulfate. So it is it needed to the plants. That's what the microorganism is doing. So microbes are the doctors to the plants, which will all the remedy giving to the plants, right? And here, biodegradation of metallic compounds and biosynthesis of the metallic compounds and biosurfactants. I told you now, biosurfactants, that the soil particles absorbed by the biosurfactants, and you have the metal ions released from the soil. So these metal ions, again, this metal ions is not harmful. It is not harmful because binded with the biosurfactants. So what is biosurfactants? Already told you, it is a balloon-like structure produced. It binds the heavy metal. Slowly it degraded. So it won't to give up called the biosurfactant. In environmental study, you may be very much study about the biosurfactant. Even the foam is formed, foaming. You have to spray the biosurfactant on the field. The foam reduced in leather industry, soap industry, chemical industry. Biosurfactant is very much applicable there. And then next is the mechanism of heavy metal transformation by the PGPR. So our core research, what it do, what did the PGPR do? That metal microbe interaction continues here by the microbial cells. How? Biodegradation of chelating agents. Metal chelates into simplest form, converted into O2 and M2 plus. That is the ionic form. Ionic exchange, degradable. That is what here I have to mention. Bioreduction, biotransformation, metal oxidized soluble and metal reduced insoluble form into soluble form. Biobleaching, what it is? Insoluble metal into the organic form and soluble metal chelators. Biopsy and all the heavy metals into the available form of the metals. And accumulation, there will be accumulated in the form of yin, it may be the heavy metal, and releasing may be the normal one. It is easily available to the plants. Then microbially enhanced chemical absorption, that is the chemical absorption of plants, hydrogen, uranide, phosphate, which is needed for the plants as a biomineralization. So the entire cycle will taken up by the PGPR, which gives the metallic form which is needed to the plants. And then here we are talking about many things in the rhizosphere. So what will happen in the rhizosphere? So if you take the root, okay, so root system, this is root system, here rhizosphere and microorganisms are embedded on the near root system, and you can take one bacteria, okay, so iron deficient environment, that's what, that iron deficient environment, wherever it there, you will have the bacteria as well as the AM fungi, arbuscular mycorrhizal fungi, and it will have the chelating, that where there will be a lacking of phosphorus, lacking of iron, it will bring to the rhizosphere. And what it will do? It will convert Fe3CD plus siderophore, enter into the cell wall and outside periplasm, inside, converted into chelation process and cytoderm, and you may get the machinery and you will get the available form of the iron. This is what the bacteria is doing in the rhizosphere. So that is the mechanism I have given here. This bacteria will do the non-available of iron into the available form by producing the siderophore. So the siderophore not only chelating the ions, all the metals, other metals which is needed to the plant, which is not available, it will convert it into the available form. And another one is the siderophore assisted bioremediation. So whenever you talk about, you have to think about the siderophores. So siderophores, iron chelating agents produced by the PGPR, and the siderophore assisted bioremediation. What it will do? Precipitated biopsy, bacterial cell, it increases the microbial resistance and metals and the metalloids bioremediation. So it will do the metals and metalloids biodegradation. And this organic contaminants bioremediation through ROS production. And here you can see the plant, it increases the metals and metalloids bioavailability, metals and metalloids phytoremediation in the plant, enhanced phyto extraction metals and the metalloids through this one, that is the PGPR, bioremediated or siderophore-chelating agents produced by the PGPR. Now, what will do the siderophore mediator? We are talking about siderophore and many pronounced in many parts. What will do? What is the function of the siderophore or siderophore? So, if you take the gram-positive bacterium, okay, in that bacterium, if uptake of protein, cytoplasmic membrane and the peptidoglycan layer. Generally, you can name it as the positive and the, that is the preliminary work, gram-positive and gram-negative. So, peptidoglycan layer, this is the structure of the bacterium, and here, this uptake of protein in this layer, you can see here, this is the complex, this siderophore may act upon the complex metal into the simplified form. So that is the covalent. This is the complex material which is here, uptake of protein may converted into the simplest form. So that is the work of the siderophore. And again, first one we discussed about the siderophore. Now we are going to discuss about the EPS. What is EPS? Extra polysaccharide. Many of the bacteria, those who are beneficial bacteria, may produce EPS. So if you want to do the work, first of all, one of the biochemical character, we have to see whether the bacteria may produce EPS or not. The amount of EPS production, we can encounter that bacteria may be the good for the plants as well as the beneficial bacteria. So this EPS not only for the plant bacteria, in animal bacteria also. This EPS is a pivotal role to protect the environment. It will protect, it will safeguard. How much of EPS production, that much of the protection given to the microbes, given to the plants. Okay, so this EPS producing bacterial cell, this is the EPS, this is the bacterial cell that is produced. This bacterial cell with metals, you see here. So bacterial cells with the heavy metals cannot enter into the bacterial cell. This EPS with metal only binding. You see here the EPS will bind with the heavy metals. It won't be taken up the bacteria. Bacteria is safe because of the outer coating of the EPS protection. That is extra polysaccharide. It is like a slimy mucilage kind of thing. Rhizobium may produce more number of EPS. Pseudomonas, PGPR, all the PGPR may produce more number of EPS. So if you want to check whether your bacteria comes under the PGPR grade, you have to check it out whether it is produced more number of siderophore. If more number of siderophore produced, first thing. Second thing, you have to encounter whether it is producing the EPS. Yes, EPS, extra polysaccharide is producing. Then second thing, it is eligible into the PGPR. Then you have to encounter whether it having the capacity of the IAA, indolactic acid production, as well as you have to check it out the quantification of the phosphate. Because rock phosphate, morning we discussed about with Castle, that rock phosphate converted into the simplified phosphate. Okay, because rock phosphate cannot take care of the plants. If you do in the experiment in your lab, that you have to check it with whether the phosphate, qualitative or quantitative analysis, phosphate solubilization is done by your bacteria or not. You have to incorporate rock phosphate on your phosphate solubilization picoxia medium. So in that medium, you have to incorporate the rock phosphate only, not by any chemical. Say PO4, it will be degraded by your microorganism. You have to put the tick. So these are the criteria you can ensure your microorganism may be eligible as a PGPR. These are all the biochemical tests we do in the lab. Then only one product will come out. So a product won't be come out by isolating and give up to the farmers. It is not like that. We go step by step. We are eliminating. Is it all the rhizobia will produce EPS? No. Is it all the bacteria, all the what you are telling the beneficial microorganism having the potential? No. You have to enhance. You have to isolate. How in our classroom we are selecting, he is the bright student, he is the dull student, he is the moderate student. Accordingly, you have to select the bacteria and fungi which is beneficial, which is good. Some bacteria may be nullified, it won't do anything. In classroom itself also, the student won't do anything. Better the 5-star chocolate, if they remains same, there is no use at all. So you have to stimulate, you have to stimulate. So far you have to screen the bacteria by producing the EPS, extra polysaccharide and siderophore production, phosphate solubilization and IAA. These are all the quality or the criteria. Then only you have to select the bacteria. Above all, again we have to, that before entering the lab we discussed again. I am using the Professor Kasim statement. I thank you Professor Kasim, I am using many words from you. I am just checking. He used, this bacteria enter into the soil, is that bacteria is compatibility with the other microbes. That is very prime important. If it is the enemy or enemy, other beneficial bacteria, there is no use, even though it produces the good things. Because one villain, one enemy may kill the entire thing. Is it not? So we don't want that kind of bacteria anymore in the soil. So we go for the eco-friendly symbiotic. That's what he has to tested his own product, ketomine. He told me, that's what I knew. I came to knew that one. Ketomine is beneficial into the any bacteria. If I want to introduce my rhizobium to his field, like he already introduced the ketomine in that soil, no problem. The ketomine welcoming my bacteria and accepting, and both are friends, like as our Axi family. All are together and we have to, hands are put together and we grow up the organic way of cultivation. That is what we need. We go, I alone want to grow, it's not a good thing. We join hand together, our imagined sound will come. You have to join. It's a thing. Again, this quoted by Professor Kasim. I am so much thankful to him, because every meeting he used to tell, I have only 2 hands. I need more hands across the globe, not his own country. Sometimes I am amazed. Are you a Thai? Are you a Philippine? Are Indonesia? Are nowadays I am asking about India. He told, I am the global person. So I have only 2 hands. I need all of your hands put joined together to take up the globe in eco-friendly manner, to spread all over the world in the organic way. That is what his motto. He started the AXI Association. I am so proud to be a part of a life member in the AXI. Okay, so that we have to think about the same way to the bacteria itself. So we have to think about either this bacteria produce EPS, siderophore, everything is that associated with the other existing soil microbes. Is it go with the friendly way or enemy way? That is simplest terminology I am using, enemy and the friendly. In scientifically we can call it as the either in the symbiotic or non-symbiotic. So if it is symbiotic way, we have to appreciate it. So these are all the criteria to screen. On of sudden during the overnight, if you are culturing the plate, next day it won't be commercialization. It not will happen. 25 years of service, we bring out one product because we keep on checking. Checking is not a single day. You have to check with the different climatic conditions. You have to the temperature, you have to the cold, winter, autumn, all season. You have to check it with all the season as well as the mostly all kind of vegetation. It should be eco-friendly. And you have to be that microbes should be accommodated in the rhizosphere. It won't be harmful to the rhizosphere because it is closely associated with the rhizosphere. So you have to encounter all those entire thing. Then only you bring back to one product. So we are dealing with the living organisms. So it is not a day overnight process to bring about one product. It is not like that. You have to keep on work. You have to keep on do how we are united in the different climatic condition, different language, different lifestyle, but we are united in one thing, that is the organic agriculture. Similarly, we have to find out the microbes to bring out into the environment as a eco-friendly manner. Okay, so this is the another criteria which will help the EPS, which will help with the heavy metals. And this is the, again, I have to tell about this EPS. It is a substances such as complex, blend, high molecular weight, microbial prokaryote and eukaryotic biopolymers. This is a biopolymers. It is not a single one. EPS is not a single chemical. It is a biopolymer and they are mainly composed of proteins, polysaccharides, uranic acids, humic substances, lipids, and one of its essential constituent is the EPS released out of itself defense against the harsh conditions of starvation, pH, temperature. Hence, it displays extraordinary physiological and physiochemical properties. So, if you have the polysaccharide, in your micro, it is a good one. And then precipitation of heavy metals by these biosurfactants. I often use this biosurfactants. Now you can see how it works. The heavy metal adsorbs on soil surface and this is the biosurfactant producing bacteria and it produces a balloon-like structure. These heavy metals bind with the biomolecules of biosurfactants produced by the bacteria and this biosurfactants start degrading the heavy metals and it binds and it gives the precipitation of the biosurfactant out of the complex. So it slowly degrade and gives to the releasing of the soil by base of the heavy metal into the available form. And then I would like to insist on some of the, so far we have seen the heavy metals and how it will be remedied, everything. I need to one or two slides. What is the, how it will impact in the human health. Okay, so human health, how it will be impact in the human health. And now is there heavy metals like lead, chromium and nickel, copper, zinc and mercury are natural elements with relatively high density compared to water. This is having the high density when compared to the water molecule. Then only it is a heavy metal, right? And in trace amounts of heavy metals such as copper, zinc, Fe are the essential of the human for numerous vital biological processes in human physiology. We should not think about map load, about zinc, copper. These are all the essential. Absolutely, this is essential. By next, you have to think about toxicological manifestation arise at even slightly higher concentration. Slightly higher concentration owing to their large-scale industrial applications like pharmaceuticals, pesticides, plastics, rubbers, chatteries, organic chemicals, wood products. These are all the sources of these heavy metals. So if it exceeds toxicological manifestation as they given, if it is slightly exists, it may cause harm to the environment as well as the human. The occurrence of free forms of heavy metals in terrestrial as well as the aquatic system is on rise because of the modernization. Moreover, the non-biodegradable nature of heavy metals ensures its prolonged presence in the environment. Their higher aquatic solubility triggers bioaccumulation and biomagnifications, which eventually leads to insights and irreversible health hazard. That is the underlying point, irreversible health hazards, and including potential carcinogenicity. By introducing our Vice President, he is given the alarming, danger alarming. Nowadays, the carcinogenicity will increase even at the lower age, younger age, because of the heavy metals. Because of the heavy metals, it leads to the cancer. That is the irreversible health hazards. That's what now we are fully, totally concentrate about the heavy metal decomposition. Okay, even every minimal concentration is range about 1 mg per liter. We should not neglect. It's a matter of mg only. Why should we think about it? That is cause the deadly, dangerous hazard ones like the carcinogenicity. It is irreversible. This is the red alarming to the globe. We have to think about how we have to rid off these heavy metals through the organic. That is more important. Again, we should not go into the ditch and the way of chemical radioactive. Okay, and the different heavy metals ions have different mechanisms of toxic interaction in infestations. Oxidative stress of free radicals imbalance. Free radical imbalance. Today morning I heard of free radical. You know very well free radical. That is toxic substances which will flow on the blood stream. So in that blood stream, these free radicals go along. It will give the senescence in our body system, and due course it may be deposition free radicals in our body system. It causes to the cancer. So you have to remove the free radicals, through which you have to consume the greens or vegetables, and then oxidative stress of the free radical imbalance, which is caused, lead is caused. Another harmful thiol or methyl derivative formation, mercury, arsenic, chromium. And another cofactor are metal ion replacement, aluminium and cadmium. Cell membrane permeation, disturbance in ion canals, DNA and protein damage. What it occurs? Chromium. Binding vital proteins and causing further discrepancies. Who's causing? Cadmium. Corrosive effects, saturation, organ penetration and lipid peroxidation, iron are some of the predominant ones. So each heavy metal is responsible for each diseases. All diseases are irreversible. That is the worst thing here we have to pronounce. If you example any one of the heavy metal, I will give the example here. Inorganic arsenic ion accumulation in living tissues. It causes the bronchitis, cancer, hypoketosis, liver crisis, mental disturbance, ulcer, all in the individuals. Sometimes we have so much of stress. Why? Because not of the traffic. By the traffic time, you may have by the traffic time, you may have linked by emitting the automobiles, which is emitting the lead pollution. That's what we have the stress. We have the tension. If prolonged, you have to think about those who are settled in the house. They are very free. Their faces are very clear. They are very happily living. But those who are going office goers, or the school goers, or the educators, or the business people, they are very tensed in their face. Because of they are exposing more number of heavy metals. That the heavy metals may lead to the free radicals deposition in our body. That's what we may have the tension, irritation. That is the simplest step. But prolonged, it may cause deadly, dangerous disease. Cadmium ions can cause a chronic toxicity leading to proteinuria and lung emphysema and upon the airborne exposure. And higher dosage cause the acute toxicity, headache. I told you now, headache, nausea, diarrhea, osteomalacia, chromium ion toxicity, making a hexavalent form, incursion at occupational level, cause the skin ulcers, while inhalation lead to determines. That's what I am telling. Because lead, cadmium, these are all every day we are seeing. Because we are going on the road, it will emit. In school studies, always we are studying about the lead pollution and chromium by the automobiles. You cannot escape. And effects of lead toxicity very widespread from the hematopoietic, hepatic, renal system, even nervous system. This exposure about 400 to 600 microgram per liter. This is the underlying word. The exposure at about 400 to 600 microgram per liter can also lead to chronic toxicity, which is characterized by persistent vomiting, encephalopathy, lethargy, tetany, these are all and leads to coma if not attended timely. These words may be the Latin and French, but we know the harmfulness given exposed to the 400 to 600 microgram per liter lead toxicity. Then think about ourselves. We are exposing daily. So what is the remedy? Go for the organic way. And this micro remedial with this synthetic biology. So we have to, again we go to the doctor. Who is doctor? Microbes is the doctor. Okay. How we do? Increased rate of bioremediation using the conventional methods, ex situ and in situ application, internal and outside. Both the application, you have to think about the increased rate of bioremediation through the synthetic biology plus bioremediation. You have the genetic circuit and whole cell biosensor, gene editing, microbial consortium, metabolic reconstruction, synthetic bioremediation strategies, functional bioremediation genes, and then you have to bring to the potent bacteria with the functional genes. I told you, there is many process you bring back to the potent bacteria functional genes, and you have to apply it in the conventional method, not in the advanced method. Into the field, you will get the microbial remediation in the soil. Then I would like to give some amount of here I have to, because we have to visit the golf ground yesterday. So in that golf ground, I have a little confusion and a doubt. Why the golf ground want to be a organic way? We need only the turf with the greenery. It's a pleasant one. And in the beginning itself, I discussed about it. But even though I need to be the witnessized, meaning he need to be the evidenced one. So, if you have to take the golf ground bioaugmentation means you have to go with the golf ground into the organic way. What you do, it's not an easy joke. It is not a one-day process to convert it to the, you can do where in the artificial turf. Okay, here is the real turf. Okay, what we do, the golf course may have the trouble in growing healthy turf. Very troubled. The turf may not grow luxuriously and easily. It is very hurdle to grow. So the site was previously used for agriculture. So this gold before you start, it is not an easy one. So you have to develop in the organic way. First of all, you have to convert the entire area into organic way of cultivation. Then only you go for the entire thing into the organic. Then only it will be worked out. The application of conventional fertilizer or Faraday acids were added to the course, maintain the aesthetically pleasing and healthy turf. Heavy metals such as copper, zinc serve as the key components of many of these products. In healthy turf, concentration of range from 5 to 20 ppm of copper, 20 to 55 ppm of zinc, and there has been increased reports in thinning turf with prolonged use of products containing the copper and zinc. Yesterday's song also detected the same. She told me about this information. So I took from her, oh, this is the amount we need for copper and zinc. If it is zinc, you go for the bioremediation. So here the PGPR based inoculants were recommended to implement the bioaugmentation, which is doing our Professor Castle, to improve the turf, healthy and applied Faraways through the irrigation system. These products contain highly diverse communities of soil, amiding rhizobacteria to reduce heavy metal toxicity and promote plant growth. So this is our important thing. So you have to reduce the heavy metal toxicity in the ground. You go for this kind of the PGPR. So it is not an easy job to convert it into the gold ground by applying the urea or nitrate or anything. No birds, even no human will come and can play in the healthier way. So if you do, you go for the bioinoculants. So that's what I think he is doing the good job. And for this evidence, I told you, no, I want to I want to prove it. Okay. So with the permission of Professor Castle, this is the video mode. Audio will come later. So here, see the researchers take down all the heavy metals, macro and micronutrients, pH, they are testing. So he is guiding us. So they are testing, they are noted down all those things. So they have to encounter whether this golf ground area may be the organic way or how to improve. They are checking the nutrients here. So here they are checking the nutrients and he is giving the instruction how to do and how to promote. Okay, so he is using that is the heavy metal, that is the biodegradation of heavy metal using his own product. So he telling about how much of the nutrients. I think if the volume is good, we can. Yeah, you can now you can tell you can see. So you can see instantly they are taking out micro and macronutrients along with the heavy metal checking. I told you now this is not an easy process. They are checking the entire ground is fully organic way. They are doing. So these people are discussing about how much of the minerals present, what are the nutrients present, how we have to improve this ground through the organic way. So everything in the practical manner. He is not sitting in the laboratory and go and apply. No, not like that. He is directly go into the ground and checking and giving the solution. Giving the solution for the that is the heavy metal degradation, that is the organic way of the product he is introducing and he is giving, and the scholars are taking down. So this is called the real lab to land concept. This lab to land concept only can achieve and we can do the research. Simply we cannot, the things what we are doing in the laboratory is not be work it out. The young researchers, those who are hearing this lecture, you kindly do your work in the lab to land concept process, and you have to improve your product, you bring back to the commercialization, and you can give the job to the others. You should not be the job seekers. You have to be the job providers. And here, this adaptation of PGPR for phytoremediation, here you have the analysis of the contaminants and choosing the microbes. That's what in previous video. First you have to analysis that the soil ground. Okay. Next, the microbes are examined, culture, placed and polluted environment. You have to check and you have to recommend the microbes, and these microbes consume all the contaminants, organic nutrients and grow enormously, and they converted into those nutrients into carbon dioxide, water and removal of contaminants occurs. So this is what the cycle we have to do. That is what you saw the previous video. And here I have list of heavy metal tolerant rhizobacteria, Bacillus subtilis, Pseudomonas, Acinetobacter, Bacillus liquefactions, Pseudomonas halicalifalia, Pseudomonas aeruginosa. So these are the potent heavy metal tolerant rhizobacteria. We have to test it how we have to given in the genomic sequences, we confirm the bacterial isolates, not the isolate as such. We converted it to the strain. Okay, and we have to conform it to the genomic sequences and the phylogenetic tree. After that, you name it as a strain, and you have to develop into the mass production of bioinoculant for bioremediation. You go for the first plate culture, and then broth culture, and then you have to formulate it as the carrier material. Then you tested in the lab in part culture. Then you go for, don't stop here itself. Then you go for land. Okay, in the land, because many of the product may success in the lab itself. Lab itself, you giving the nursery. So that is called the nursery. You giving entire, it's a good environment. How the baby is crawling the mom's lap. Okay, it is very good and very sophisticated. When it crawling on the ground, it feels the knee pain and leg pain and started to cry and maybe fallen down. Okay, so that is what here you have to given to the application in the field level. So it may be have the all kinds of environment, rainy, drought, or anything. So you have to expose it to the field. Once you achieve in the field, you go for the industrial type. And this is my, this is not 20 years, now it is 25 years of my research work. I started my career in 2000. Okay, as for in that field of what is that, organic agriculture, and then by tracing and Almighty and Professor Castle, we went to here China in the Songyang province. Fully they converted into the organic way of cultivation. That too lead by our Professor Castle. That's what we went. And another important story I want to tell here, in that PR China, they are not inviting me just like that. They given the invitation like a red carpet. Red carpet invitation, entire Songyang province, they given in the poster, topmost banner. Why? Only of organic way of cultivation driven by our Professor Cassie. Nothing else. They want to convert it into the Songyang province into the organic way. They given the scientists with the red carpet welcome. Why I am insisting here, it's not the faming or our appreciation. We are not, that is the faming go behind. This is what the effect of the organic form. That's what I want to insist here. Okay. And here within our lab, we are not doing such a namesake. We are not doing. Every year we are conducting. This is our Vice Chancellor, and these are all the farmers, and this is my scholar. These are all our products. So we are doing every year with the farmers. We have the discussion what are we are doing in our laboratory. It's not a simple job. We are doing continuously. That is called we are doing the sustainable agriculture. I think with that note, I conclude myself. If you have any queries and clarifications, you feel free to talk. And in the first slide, I'll give my email ID, and you can reach you at any time. This is my email ID. You can reach you at any time, wherever in the globe. You can contact me if you have any doubts or research collaboration or anything else you want. You can contact me, and I would profusely thank my authorities in my university, my Indian authorities, the Vice Chancellor and Registrar Sir and my head of the department, because without them I cannot come over here and they permit me to present this function and present this talk. I profusely thank my authorities and I profusely thank the Thailand government, and my special thanks mentioned to Professor Castle, who is the backbone of this event. This event is going very well, and for this note, I would profusely thank Professor Castle, and if I fail to thank my scholars, I have not justified myself, because the entire thing, as Professor Castle sometimes said every time, my hands is two hands only. I have to collaborate with my scholars only. I have to bring about this kind of research programming. So I would like to thank Uma Maheshwari, Shakti Uma Devi, and Sridhar and Satya and other my research scholars, previous scholars, present scholar, future scholar, and my PG students, who is maybe they may be the observer for this meeting also. I would like to thank my scholars, my faculty, my colleagues, everyone. Thank you, everyone.
Okay, give a big hand.
So, thank you very much for both Lalita to give very excellent presentation. This is the very real knowledge. I think you are very good, and I am more excited all the time. 1.5 hours or 2 hours. Nobody sleep, And then I think our participants from the online international training for the organic tropical fruits will be hear us, will be know about what we have done. The participant is over 50 countries. So we would like to welcome all participants from about 50 countries all over the world to hear about what we have done in a corner of Thailand and India.
Yes.
So this is the what we talk about. That's why we say that this is a modern of organic agriculture. And this word I give this word for the first time in China. In the year of 2015, that you also, at that time we have the international symposium for the modern organic agriculture. And at that time, we organized by Songya government, China, together with the Association of Agricultural Technology in South Asia. And that time I invited our group of scientists that we are doing research on the organic agriculture. About 18 countries, 18 countries, you remember. So that is, after that, everybody, we try to concentrate to do research. Especially, in my heart, I try to help society to be with good health, especially human being. And I always say that I have only 2 hands, I need more hands to help each other, especially young generations.
Yeah.
So this is what we have done.
Yes.
Thank you also support. Thank you. So this is the Dr. Lalitha presentation is very active and very practical work. So like for example, what you have the idea? For example, I like Dr. Pierre from Cambodia. You expect something. This knowledge for apply the microorganism to reduce heavy metal to apply in golf course. What do you think about this? You expect some idea? I like to comment about because Dr. Pierre will do the postdoctor in the organic golf course.
My name is Pedro. After I listen to the presentation, I think that the microorganism, like some bacillus, can be applied in the copper to reduce the heavy metal in the soil in the copper. I will do this research in the copper, and you will see the result later. Thank you.
Okay, thank you. Dr. Pierre is the postdoctor at King Mongol Institute of Technology, and he's from Cambodia. So another one that I also very interested in, Dr. Lalitha presented about how to reduce the heavy metal in hydroponic. Actually, if we are going to do hydroponic, you know hydroponic, the chemical one. I never eat it. You know why? Firstly, I found a lot of nitrate.
Oh.
Yeah, a lot of nitrate. And in the in hydroponic, also nitrite. Both nitrate and nitrite cause cancer.
Yeah.
See, and also I think your presentation is really good. Your experience is really good. That heavy metal may be contamination in water,
Yeah.
in solution,
Yes.
and after that, not only nitrate,
Yeah.
but also heavy metal.
Yeah.
So this is we have our student from Philippines, Sernan. He's worked on the research on the organic hydroponics. Sernan, you come to make a comment. How we apply this knowledge to your research? You express your idea.
Yeah. Thank you very much, Dr. Lalitha, for that wonderful presentation and encouragement and for awareness about heavy metals. According to my research, I'm doing research about hydroponics, and it's enlightened my research now to really upgrade or update because you mentioned about the heavy metals. And I think in my research, if I do the organic way, I can really help encourage on how to do the sustainable agriculture. Thank you very much.
That's great. Thank you, Sernan. Sernan is from Philippines. So I think today you all participants when we get more knowledge. This knowledge is for our experiences by research,
Yeah.
and that's why if you are going to do the organic fruit production, and is this one factor is very important that this you must concern about heavy metals, that contamination in soil, in water, how to get it off, or how to decrease heavy metal lowest as much as possible to be the safety level. And this is one of the very important factor, and that SC or SC do organic certify, because SC whenever SC member or SC organic inspector going to do organic certify, we concern about pesticide residue. We concern about nitrate accumulation in soil and water, and also heavy metal also, and human pathogens, Salmonella, E. coli, and others. This is our regulation. That means this is very important. If our participants they try to or interested, please don't hesitate to contact us. And we are here in our world, the same world.
Yeah.
We are here, but different corner of the world.
Yeah.
So this part, I hope that everybody will get the knowledge about this one. This is one of important to do organic fruit production. I like to confirm again. And I heard something, we have been checking the durian fruit.
Oh,
You see, and the durian fruit have the over heavy metals.
Oh.
So if durian fruit over heavy metal, we will never certify. Even not detect pesticide, ND, not detect nitrate,
Okay.
but over heavy metal, how can we certify to be organic?
Yeah.
Durian, we
Yeah.
not certify.
Yeah.
So that means if you like to get it off or to decrease this one, we can help. We can help by scientific way.
Yeah.
So I hope the all participants that you are interested to do the organic fruit production any part of the world. That, because participants now is from 50 countries.
Yeah.
Any part of the world, you can contact us.
Yes.
We are the same world.
Yeah.
Don't worry. We are the same world. We can help each other to help our world, and at the same time we can protect our environment as well.
Yes.
So you talk something to our participants,
Yeah.
please.
Yeah. once again. And if you have any queries, you contact Dr. Castle and also myself. So feel free to ask, because the fruit is very much essential. It is a second staple food. Nowadays everyone, the patients, infants, that is children, everyone is taking the fruits and fruit juices. So it plays a pivotal role. That's what this online course mainly emphasis on the fruit, tropical fruits. Training course on tropical fruits. Because why we have to call it this name? The fruit is the staple food for everyone. So you have in the fruit breeding in the organic way, in the free of chemicals, free of heavy metals. It will be give the best contribution to the society. That's what our motto, to give the organic way of fruit. It's a good for health. So you have to improve the health. So that is the contribution to the society. So with that note, you have to utilize this kind of program. You have to keep on listening all the resource persons. They are taking effort, much effort, and giving the lecture. And you have to utilize the young researchers or the beginners, mid-term careers. You have to take up the points. Some point you may have triggered, and it will turn up your lifestyle, your research. So you have to take up the words, you listen, you hear all the available resource persons talk, and you have to improve. Best of luck.
Okay, thank you very much. So today I think one of you have the listen about the how to reduce or how to degrading the heavy metal in the soil. This I said again. This is one of the important for organic certification. And I would like to thank everybody. And actually, our training, online training for organic agriculture is about for the organic fruit production. We start from June 4 to June 16. So I cannot live all the time, and in different countries the time is different also. So that means some part I will live presentation, and some part I will do record video, and like this record video, and then we can contact each other. So even during this internal training, someday I will be in India also, something like that. I also move around like that. So anyway, at least you know what we do and what we can help. The Association of Anatomical Technology in South Asia, or SC, is non-profitable organization, and we try to build up the society to be safety environment, and also the young generation grow up without heavy metal. To be like that. So I hope everything will enjoy our training, and so thank you very much, and then see you next time. Thank you very much, everybody.
Thank you all. Bye.
Bye.